honor to introduce our third keynote speaker, Dr. Palati Alish Sinu, Associate Professor at Department of Zoology, Central University of Kerala. His impactful work has significantly contributed to the advancement of life sciences, biodiversity, and conservation. Dr. Sinu brings with him a wealth of experience, and his remarkable work has earned him a national and international recognition, making his presence here truly valuable for all of us. It is now my privilege to invite Dr. B.R. Jyotode sir to formally introduce our distinguished keynote speaker, Dr. Sinu, and share with us a glimpse of his inspiring academic journey and impactful contributions. Over to you, sir.
Thank you, ma'am. Good afternoon, everyone. Myself, Dr. Vilas Jyotode, shortly introduce the speaker of this session, Dr. Palati Alish Sinu. Dr. Sinu is working as an associate professor in Department of Zoology, Central University, Kerala. His area of research interest are tropical terrestrial ecology, biodiversity, conservation of forest landscape, insect-plant interaction for pollination. He has more than 15 years of teaching and research experience. He has published 3 books in Springer and 100 plus research papers in reputed international and national journals. In last 5 years, he has published 91 papers and in his name with 22 H index, 42 item index, and sir have total citation 1300. He guide 7 PhD students, 2 MPhil students, and 72 MSc students with 1 postdoctoral student. He has certain achievements like Royal Fellow achievements in United States. With this, I also felicitated here the Professor Dr. D.D. Barsagde sir, former head, Department of Zoology, Rashtrasant Tukadoji Maharaj, Nagpur University, Nagpur, who worked for peoples, train the peoples in sericulture and provide platform for employment to peoples in sericulture. Thank you, sir. Now, I request to Dr. Sinu to start the lecture and guide us. Thank you.
Thank you, sir, for your outspoken introduction of Dr. Sinu. Your words have vividly portrayed his extraordinary academic journey and the profound impact of his contributions to the scientific community. As we forge ahead in our pursuit of scientific discovery within a vast realm of life sciences, get ready to be deeply inspired and intellectually enriched by the insights of Dr. Sinu. His research has consistently pushed the boundaries of current knowledge and inspired countless minds. I encourage everyone to remain fully attentive and open as his insights are truly to ignite new ideas and perspective to all of us. It is my great pleasure that we now invite Dr. Sinu to share his insightful perspectives to broaden our horizons. We eagerly look forward to gaining valuable insights from their address. Over to you, sir.
Thank you very much. I hope you can see my screen.
Yes, sir.
Yes, sir.
All right. Thank you, Ankita, and thank you, Dr. Vilas, for inviting me. And his guide, Dr. Deepak Barsagde, was pivotal actually to bring me into this very important international seminar, one-day seminar. I congratulate both plant science and animal science for organizing this conference, this seminar. And it is very important to discuss the biodiversity and its all ramifications and its ecosystem service roles. I'm happy again that the 2 departments are jointly organizing this program. It is wonderful because every time most of those departments want to keep themselves in their own identity. I'm happy that the 2 departments under the aegis of biology are offering this seminar. All right. So I'm as Dr. Vilas and other introducer was talking about I'm an entomologist basically, but after my PhD, I was really fascinated by the research of the pollination and pollinator biology studies from Professor Shivanna. He is considered as the doyen of plant reproductive biologist. So, in a way, he really baptized me to the world of plants and intricacies in the plant pollinator interactions. In fact, he had a DBT project long back about 2012, no, 2006, and he was looking for an entomologist to help him in his project. So that is how I moved to his project. Since then, I'm working very actively on plant pollinator interaction studies, very complexities in the interactions. With this introduction, let me begin. So this is, as you all know, a plant pollinator interaction is not only in the field of biology, but also in the field of many other dimensions, including the social studies, including economics, including commerce. It is a very important ecosystem service, right? If you're going to value the service, it is trillions of dollars in Indian rupees each of the producing countries are getting through this interaction. And in that way, we are also evaluating the services provided by both the manager as well as the wild things that we see in our surrounding. When the developed countries are already prepared to manage their pollinator conservation, unfortunately, India has taken it as granted that they are already there. We don't have to conserve the pollinators. So I'll be beginning this session with uh, some textbook information for the students, anticipating that they are there to talk about a little bit on the plant-animal interaction, and I will be talking about two crop species what I worked for the plant pollinator studies and to highlight the need of conserving pollinators. So as you all know, students and also other members of both plant science and animal science and beyond, we all know that interactions that we see in the human community, we can also see this kind of interactions in the whole world of biological systems. So what are those broad interactions? So one is we can group them as positive interaction. Another one is a negative interaction. Nowadays, we don't have true friends, right? Every friendship is just for bargaining things and also to get out of something, right? That's what I used to say in my classroom, right? If one is approaching you, there is no selfless friendship, but we can see some of this selfless friendship in the world of biology. But again, we cannot universally generalize that statement. So in positive interactions, we have mutualism. So if you are giving a textbook, I will be giving my resource. I will be sharing that resource with you. So that is a mutualism. Both of them are commensalism. I'm not harming, but I'm not ready to give what I have, but I'm just there, right, to facilitate you. But in negative interactions, we have herbivory parasitism and competition and predation are all come into the world of this negative interaction. If you ask me what is the role of negative interaction, mainly the stability of the ecosystem, we need both the positive and the negative interactions. If you go and browse the newspaper articles, or if you're going to talk to these reporters, they want to know what is controversy in it, what is the negative trends happening in the world. No reporter is very happy to glorify the good things happening in the world. They're all behind what is happening and negative things happening in the world. But I want to tell that has its own role. Criticism, so people think is useless in the present world, in the present political atmosphere in the world. Criticism, whoever criticizing immediately will be phased out from the system one way or other. But in the world of democracy, in the world of human connectedness, we need this criticisms also there. In that way, both the positive and negative interactions maintain the system well. So much so that every partner or member involved in this interaction will be in the part of that ecosystem will be thriving their populations. So if you look at this posit interaction, as I mentioned, there is a partner, there are partners, a couple of partners. If you go for one-to-one mutualism, so a partner species provides food, shelter, or substrate to grow, or onto one other partner, it may transport its genes either in the form of the pollen or in the form of the seed. In either way, it is nothing but the reproductive parts. So that is another way this in the world of mutualism, if you can see the interactions going. May also decrease the negative effects of competitions and herbivores, predators, and parasites. Some way that this facilitation will be always highlighted than those competitions. But as usual, as I mentioned, we cannot generalize this mutualisms are always a mutualism. There are cheaters also there. One of the articles that we recently published in the Journal of Oikos, you can see the picture of Lucas aspera here. And Lucas aspera, you may be wondering, in the left-hand side picture, there is a one Lucas aspera looking very different, right? There is a fissured Lucas aspera flower. On the right-hand side, in the left side picture, you can see the intact Lucas aspera flowers, but there is a flower on the left extreme left. Yeah, isn't everything fine? Yeah, so there you can see that the flower has been defaced or it has been damaged by somebody. So in our research, we found that it is nothing but a bee is doing this job, and that solitary bee we call haplonomia in the family of this Halictidae, and in the nominae subfamily, you can see that bee is in the way of getting the nectar, right? Because this flower is not really good for her or for him to have the nectar because this traits of the flowers are good enough for a bee which has a long tongue. But the bee is very deliberate to have the nectar from the flower because it is so sweet, and she also might know that Lucas aspera in the world of human beings is a medicinal plant by I sip nectar. And what she learned, the mechanism of cheating this interaction, and she started making a tear, right? And she torn the flowers and defaced the flowers and had the nectar. The question there is what kind of implications it has on other pollinators, right? Because Lucas aspera, if you go and watch in your own garden, right, you can see minimum 20, 30 flower visitors will be visiting this Lucas aspera flowers, right? So the question we asked in that question, how the nectar robbing behavior of this haplonomia species will be affecting the legitimate pollinators. The main meaning of the legitimate is the loyal friends of these flowers, because there's a landing platform, as you all know, this is a zygomorphic flower. There is a landing platform in the lower keel, and that is where the bee should sit and insert its mouth part to have the nectar. But you can see that landing platform has been damaged. So the research found that a lot of these legitimate pollinators deserted such flowers which were robbed. So that just to tell that there is mutualisms also can be compromised at some point when we think about this exploitism mechanism. So the mechanism here is an exploitation because B has so many other resources, but maybe because of the paucity of the resources, B might have learned a new method to approach a flower to have the nectar, which the flower is not actually useful for or is not made for as a partner for this bee. So to tell that the partners in case of mutualism, we have plants and animals involved, and the rewards in the case of plant pollinator interaction, so it's a pollen dispersal is a reward that plants get and animals are getting the resource that is giving the energy requirement or energy economics of the animals is depend upon what is the surrounding. So cost involved, it's vice versa for the two parties involved. So what is the floral isolation? If a flower is isolated, right? It's a visit, so it's a legitimate partner. So we call them it as an isolated, right? We also face something like that, right? When we don't have any friends nearby, we are in the solitude, mode of solitude, right? So in the world of biology, right, every creature is creatures' mission, including human beings, is supposed to be to procreate, right? Improve their fruit set, improve their populations. So that is the only requirement they have. That is the only job. The creatures, if you go and read the Bible, the creatures are created just to glorify the God. And how we glorify the God by increasing the population. So some communities in the human populations, right? You can see such is taken in ditto, and they increase their population. Likewise, the animals and the plants also doing this kind of procreational activities. But just imagine one flower is never getting a visit of its legitimate partner. What will happen to that? So plant scientist people will say that the plant can move into the direction where it can go in apomictic ways, or it can go for a self-pollination way. If it was a cross-pollinated flowers, it can switch to any of these extreme ways of fruit set. That is what happening, right? Is if you look at the dioecious plant, dioecious plants means there are two plants. So one is male plant, another one is female plant. Again, the females in the reproductive isolation because the partners the pollen should be brought by the pollinators, and the pollinators, if it's missing, then she will be in the reproductive isolation. So how to improve the fruit set? She herself will be producing small proportion of bisexual flowers in the female flowers. So in the female tree, in that way, there will be some selfing occur, and some fruit set is better than nothing. So, as I mentioned, there are morphological isolation due to the flower structure. That is what happened in the case with the Lucas asperatus, as the haploinomia species, and the haploinomia species, the find the plant is not suitable, but he learned a mechanism to exploit the resources. Pathological isolation due to the pollinators' behavior. So the mismatch is the reason why there is an inefficient pollination is happening. So if you go and search in the literature, you cannot see much of this co-evolved interaction in the world of this plant pollinators. If you will be hit by this case of the ficus, right? There are fig plants and the fig wasp. So that is one in the yucca and yucca moth interaction. So very few such one-to-one co-evolved interactions there. But if you go and vastly into the world of this complex interaction, so you can see that there are one plant is benefited by so many species of these. So more complexity in a network is good for the plants and good for the animals. Less complexity will be leading to the system what we call the specialization or specialist pollination. So I don't want to disappoint animal scientists over here, because animals also very important. They should know also their requirement, their biology as well as their ecology. So what do you look at? If you look at one primary requirement is to build on, right? What are the flower visitors, at least to the crop plants, what you're growing? Unfortunately, for the Indian crops, for the Indian jurisdiction limits, we don't have even the data. Very unfortunate, students are not being driven, at least by the college curriculum and as well as the university curriculum, to introduce the topics related to the ecology and applied ecology aspects, to bring the students to the field and to at least develop a database on at least on this biodiversity in your locality. And if you go with the functions like the pollination, seed dispersal agents, herbivores, etc., are all the world trend research happening around the world. But unfortunately, India is finding that is in the least scope. But if you look at that, you can see the insects are majority of the guild constituted by the pollinators, and birds are the other ones. So we have sunbirds around us. A good example to show, but there are so many other birds also involved in this pollination. There are a lot of mammals, including snails include, and there are whatever which can transport the efficiently transfer the viable pollen grains into receptor stigma. We call them as the pollinator, and they will be uplifted from the class of those flower visitors. So if you go and look at this literature, right, you can see that the Klein, Alexandra Media Klein and her team in Germany was looking at the literature and did a meta-analysis to look at what percentage of the crops that we consume are pollinator dependent. So it is a proceedings of Royal Society B a journal article way back in 2007, and now still we follow this literature because not much change occurred in the proportion of plants that are pollinated. You can see that 77% of the crops that we consume, and I suppose that this will be increasing because our food habit is increasing, changing. Earlier, we were consuming a lot of grains, but people want nowadays more corn, and the corn alone is not sufficient, and they want the choco corns, and there will be coconut, and they want to spice it up with a lot of cashew and a lot of other ilachi, electria cardamom, and many other things, which and fruits, etc. So if we go and count each of these constituents that make into a meal for you in the breakfast, you can see that we are more moving towards a more depending on these pollinators than years before. Earlier, our parents consuming only the grains all the 3 times a meal. But nowadays, you can see people are consuming a lot of fruits because of our health habit and other health-related issues. We now switch to the vegetarians, and the vegetarian too into the nuts and other things for the good cholesterol. And if you look at the 77% crops, over 90% of most of the fruits and vegetables are coming from the tropics. So we have an inbound job and the duty to increase the fruit set in the crops what we are cultivating, because we are the suppliers of most of these crops of that world eat today, right? And more than us, the Western people eat bread butter as well as corn meals, right? And fruits, etc. So we are again in the world of Trump, and the Trump is imposing his taxes on us, increasing, doubling, or tripling the taxes, and the countries are competing, and it is our job to maintain our population, I mean, our market, right? So, because we are producing part of the world, we should now be very careful to now increase the crop in a sustainable way. And one of the elements of this crop food security as well as the crop production is maintaining this pollinators. But unfortunately, we know very little on the pollinators in the Indian system, and we know something from the agricultural as well as the basic science universities researchers happening, but we don't know what is the trend of this fruit set. If you go and search for the ICR reports, they will be showing the statistics of what is the trend of millets, what is the trend of sorghum, what is the trend of rice, what is the status of horticultural crops. You will be seeing that is decreasing steadily from year to year. This is all showing that there is a pollinator dependent crops production. So if you look at the phylogeneity of these insect pollinators, constitute more than 90% of the pollination happening in the world. The bees are the majority of them. The reason why this, because they are very diverse, they are everywhere, they are abundant and diverse, and their functional diversity is very good to go for even one-to-one interaction between flower types as well as the pollinators. So we have very few long-tongue bees and a large number of short-tongue bees. So that is very important. I'm not going into very details of this biology of this and this interaction in that way. So my job now is to show how these pollinators are important for food security. As I mentioned, I will be showing 2 case studies. The first case study is coming from the world of cardamoms. In this picture, I have shown the picture of large cardamom, which may not be very familiar to many of you, but maybe for plant science students and the faculty there, they understand it's a zingiberasian plant and growing in the northeastern belt in the Himalayan stretches. So our research was happening in Gangtok and Darjeeling region of India, but it is also growing in China and Nepal and Bhutan, etc. So it's one of the commercial crop, particularly for this tribal belts, because they have the land inside the forest and they are allowed to cultivate this large cardamom. This crop, what we call this Electria cardamom, it is a small cardamom and which is very familiar to you. I don't know whether Maharashtra grows this, but you can see the small cardamom growing in the belts of southern region and the Western Ghats mountains. And they are different. So the questions are what we asked in the research. cardamom plants are specialists in pollination because they are showing the traits like zygomorphy is involved, right? Zygomorphy is involved in the case of cardamom flowers. Does it be literally a cardamom or it's a large cardamom? You can see the flowers are zygomorphic. So let me tell you, compared to the actinomorphic flowers, the evolution of zygomorphy itself implicates that is leading to specialization in pollination. So we asked whether there is specialization evolved in the case of this cardamoms. If it's so, which crop it is that is the pollination a constraint? So we have done this research together. So there is a entomologist, a zoologist working, and a botanist working. Professor Shivana, you can see on the right hand side. And on the left hand side, I'm lying down on the ground to record the pollinators of this large cardamom. And we studied it together. That is the reason why this project became very successful, because of this collaboration between the plant reproductive biologist as well as the animal ecologist. So we did this study in the Western Ghats and in the Eastern Himalayas. If you are interested to look at the natural history of these two plants, we can see they are cultivated in two different belts and evolved in two different regions. If you look at the small cardamom, that is Elytria cardamom, which is more commercially important, which is of more medicinal value because the oil production is much higher in the case of cardamom, and the value of that oil, edible that oil, essential oil, is qualitatively more superb than that of the Amomum subulatum. Whatever it is, they are all cultivated in the humid and cool weather of the mountains, right? Both of them are perennial herbs, and flowers are produced on long racemes in case of Elytria cardamom, in case of Amomum subulatum on short pedunculate spikes, and the white color in case of cardamom subulatum is a yellow color. Bilobed anthers in both the cases, the nectar tubes are quite long. It again indicates that this pollinator should be a long-tongued bee. Short-tongued bees and short-tongued visitors cannot access to the nectar, but maybe to the pollen that is possible. So if you look at this map, you can see how I'm using this opportunity also to educate the students who are interested to take up the pollination studies. You should look at both animal aspects as well as the plant aspects. As I was just talking here, to understand the natural history itself is a very important step to frame a study, right? To plan your study for pollination. And also you may have to look at the flower architecture and the flower behavior across the period of time. As you have seen in this picture, what I have shown in the yellow color is the case of Amomum subulatum. What I have shown in the magenta color here is in the for the case of Elytria cardamom. What I had shown here on the x-axis, I have plotted time, and on the y-axis, I have shown this distance between the endostigma column and the lower labellum of the flower, right? Why it is important? Because this is the very important from the pollinator's perspective. Because pollinator has to. Okay, I'm coming to that in very detail. Well, from the graph, we can understand that in a moment subulatum, the distance of what you see between this lower labellum as well as the upper and the stigma column, in the case of Amomum subulatum, it is increasing, right? As you can see from this end of the scale to this end of the scale, you can see that is increasing. But the reverse is happening in the case of Elytria cardamom. Earlier it was in the white, now towards the noon, it is decreasing. I will be talking about that in much detail, why it is important later. So if you want to see very clearly the pictures of these flowers in the noon as well as in the morning, in the noon for the case of Amomum subulatum as well as Elytria cardamom over there, you can see this is how this Elytria Amomum subulatum looks like. You can also see there's a long nectar tube. Nectar accumulates only up to here, compared to this Elytria cardamom. Also, a student, if interested, a researcher, should look at also how this anther is looking like, how this pollen is released, or anther dehiscence occurs, and where is the stigma, right? Whether there is a stigma, what are the mechanisms the plants have to reduce the self-pollination as you all know that the self-pollination may lead to this inbreeding depression compared to the cross-pollination. So almost all plants want cross-pollination to happen for the genetic variability in the system and also for genetic improvement in the system. So if you look at this Amomum subulatum, this is a longitudinally splitting endostigma, and there is a long style passing between the lobes, and you can see the stigma open right over here. Similarly, in the case of Elytria cardamom, small cardamom, but the difference you see in case of a stigma of the Elytria cardamom, the stigmal cup is open towards the and the stigma column. That means it can attract or it can bring the pollen grains quite easier than the case of Amomum subulatum, where the stigma column is, you can see very clearly in this picture. This is how the stigma of Amomum subulatum is projected outward without giving any single chance for the self-pollen to come to stigma. But in case of Elytria cardamom, you can see the stigmal cup. I'm not sure whether you can see it very clearly. Then you can see that is projected towards the endostigma lobes. So this is how a very sufficiently pollinated flower look like. You can see this and the stigma cup is full of pollen grains, and the stigma cup in the case of Amomum subulatum, large cardamom, in case of Elytria cardamom, the pollen grains is stuffed in this. So we asked the question first, what is the pollination ecology of the small cardamom and subulatum. So we go and this is not a workshop, but you should know that you should have a time schedule, and based on the dehiscence, you will be watching flowers. As a field practical, you can do that. You have to identify such plants in your locality. Sit with the flowers for some time with all the sunscreen and all. You don't have to worry about this sunlight during the early morning, so you can sit with them and just record the flower visitors. And also you should record what is the number of flowers you are watching, because that is used to identify what is the visitation rate in the system, right? So if you look at this, we had spent many days and many months and many years. This was a 3-year project, and we had seen that Elytria cardamom, small cardamom, has a lot of flower visitors, but Epicerena, that is a honeybee, is the most common visitor. But in case of a large cardamom, you can see the Bombus emeraldinus, the bumblebee, which is restricted, which is a temperate species, right? Which is seen only in the temperate belt. In India, it is seen only in the Himalayas, as well as the mountains of this Meghalaya is the major pollinator. So you can see the flower visitors, how they are taking the nectar. You can see in the case of Elytria cardamom over here with taking the nectar, and by lifting the endostigma column, and the Apis serena is doing here, and Apis serena is doing for this this Elytria cardamom. So if you look at this, we also see some short-term bees visiting the Elytria cardamom. So the story is now I'm focusing is on the small cardamom. You can see the Seratina hieroglyphica, which is again a very common, you commonly might have seen, or you can identify it as a yellow-banded bee. This is something what we commonly call, you can see it is carrying a lot of pollen grains on its body, as well as on its scopal apparatus on the leg, on the hind leg is taking. So that, and also it attracts large bees. They all are very important functional traits, right? So in a workshop mode, we can teach you what aspects you should look at, because it's not a workshop. I'm just skipping all this. So you should look at what is the size of the bee, intertegular distance, etc., and what is the length of this the tongue, and their behavior, times it visit on the flowers, etc. All these are very important traits to explain the function, what we call the pollination, right? And it also attracts some of the pollen foragers. So earlier I was showing all of them going for the nectar. We can see they're lifting the endostigma column, and maybe accidentally they are trapping some pollen grain on its body, on its head. But you can see some tiny bees like the stingless bees, so what we call the tetragonula or trigona species, and which is also quite abundant in the case of a small cardamom. She knows her limitations and she is not trying to cheat the flowers like the Aplonomia did for the Lucas aster, and she is concentrating or focusing on the pollen grains. She doesn't want to take the nectar of the flower. So if you look at the wild population, because we identified the wild populations for the small cardamom in Idukki, right? The district of Idukki in Kerala, that is the place of origin for small cardamom. We are surprised to see that the pollinators in the wild populations are primarily the solitary bees compared to the social bees like the honey bees and trigona or the stingless bees that we had seen for domesticated bees. It is also important to look at what is the fruit set and what is the pollen transfer in each of the visits of this visitor. So this graph, what I have shown here, is the pollination efficiency, which I define like whether that is transporting the pollen grains to the stigma. And in the red color, the bars shows the fruit set and what percentage of those flowers setting into the fruit. Sometimes both the metric is important to analyze what is the efficiency of each of the pollinator. You may be looking at this part of the graph. You can see that Epicerana nectaring, Epicerana polleni, for pollen collecting, Epicerana collecting both pollen and nectar. You can see their contribution into the pollination efficiency as well as the fruit set is tremendously varying. The point I want to make here is that some researchers may be just looking at flower visitors on the flowers and write an article that is the flower visitor as well as a pollinator. But you have to keep in mind that the pollination is not that easy job. There is self-pollen objected by the stigmal surface. So you cannot see the pollen germination at the stage, and each of the behavior is very important to incapacitate the pollen grains, sometimes to facilitate the pollen grains. Just a behavior of their raging itself is showing that the pollination efficiency across. So the second question we asked, because we had identified this wild populations as well as the domesticated population. As you all know, when we domesticate a crop or a domesticate an animal, we will be increasing the productive traits, right? Improving the productive traits. So what is productive traits, which is important for our yield? So in case of cardamom, the yield is nothing but the berries, right? The fruits what we get in the seed from seed production is the yield trait. So what we do when we improve genetic, what we will do, we will be making the new varieties from the wild ones, and the number of flowers produced, the number of clumps in the plants, all the flowers. So if you look at this, we had two varieties of domesticated cardamom here in Yelani and Mudigere one, and we had a wild wild crop that is grown up exclusively in Mona. You can see in each of this productive trait is much lower in the case of a wild case. So the question we asked whether that is the reason why this flower visitors, what you see in case of this wild as well as domesticated varieties are different. So you can see how their populations look like. On the left hand side, what you see is the crop in the wild system, where you can see much of seedlings of other plants or much of leaf litter covering all these flowers, and the plant themselves are very small. Number of flowers produced is very less. But in a plantation, if you go, you can see this varieties improved, large clumps growing, and a lot of racemes, a lot of flowers out there, all produced. Right? So you can see in this case, the domesticated case, you can see honeybee is the predominant visitor, but in case of wild population, it is the wild solitary bees like the megachilds and the amegilla, etc., are the major visitors of the flowers. So we were wondering whether this is different, the pollinators different in these two crops, as we were expecting. Because of several floral traits, we can see most of the wild flowers received visits of these wild flower visitors like Megachilamic in the species 2, like that, and their contribution to the flowers, pollination, and the fruit set is much higher. If you look at the domesticated crops, you don't see any of these flower visitors to the domesticated crops. which had a different set of flower visitors, and their contribution to the fruit set as well as the pollination is much different, right? So the point I want to conclude with this is that you can expect a difference in the diversity of pollinators and their role in pollination in case of domesticated and non-domesticated crops. When you come to the case of this large cardamom, that is another crop we looked at. We want to ask what are the pollinators, whether there is any specialization involved in this case. We can see there are honeybees are visiting the flowers at too many numbers because they are maintaining a lot of honeybee boxes. In fact, the researchers from the spices food advocated the people to maintain honeybee boxes in their large cardamom plantations in the Himalayan belts because of poor knowledge, right? Because they never knew that honeybee is not a very good pollinator. The wild pollinators of this crop is bumblebees. So you can see on the left-hand side, you can see the queen bumblebee with a lot of dust of pollen grains on the thoracic hairs, and the worker of the same species on the right-hand side, you can see carrying out nectar foraging. So let me tell you, no bees deliberately doing the job of this pollen foraging unless they want, right, for carrying to their nest as well as for the ospreys, right? Primarily, the adults are interested for nectar consumption, but accidentally they will be carried by or they will be covered by the pollen grains, which is what the plants will be using for pollination. That is how the flowers architecture came up. You can see here why you can ask the question why the stigma of this large cardamom is designed accordingly that is on the upper keel. Why it is not in the lower keel like what you see in the Papillonation flowers or the Fabaceae plants, right? Where the stigma you can see in the lower keel. The reason could be the pollinators which are coming for that large cardamom has hairs on their body can carry a lot of pollen grains and the stigma can be stuffed by the cross pollen when it is moving to the next flower. So the stigma cup locates at least 3 millimeter away from and there's a tip, so it will be stopping the self-pollination to. So a pollen collecting honeybee is not cheating in this case because she can't forage the nectar in case of large cardamom because her mouth part is, even though she is a long-term bee, her mouth part is not long enough like a bumblebee to reach to the nectar, which is way down here, right? She cannot reach. But at the same time, she doesn't cheat the flower. She will be limiting her visit to carry the pollen grains and to the nest. So as I was talking, the distance is very important between the endostigma column and the lower keel of the flower. Biology. In the morning, if you see this bee in the morning can carry a lot of pollen grains and can touch the endostigma column. But if you look at this at noon time, even the queen bumblebee, which is considered the larger one compared to the worker bees, she cannot touch this endostigma column by the time due to the process what we call the plant science people may be knowing that a flexi-styling, right? The style is moving, right? The style is moving across the period of time. So that is again one of the fine-tuned mechanism to limit this unwanted or inefficient pollination as the time progresses. So we found that the bumblebee is the pollinator, but the question is that is giving a clue that whether that is a specialized one. So we went about and found what are the pollinators in the system. We found in the Latria cardamom there are so many, but in case of large cardamom there are only very few. And if you look at the efficient pollinator, we want to identify from a very few number of bees what and the flies and the moths attracted to the flowers. So we did that in multiple populations on multiple sites in Gangtok as well as in Darjeeling, and we found the percentage of open pollination efficiency. What is that? Open pollination efficiency means you are bagging the flower a day before it is opening and keep it opening on the day of opening, and in the end of the day you will be taking some flowers and examine under the stereoscope microscope and you see how many of them have pollen grains on the stigma. So we find that the percentage of open pollinator flowers is varying a lot between populations, right? Why so? So we were just looking at what are the visitors and what is their efficiency. Look at this table. The Serrana, that is considered honeybee, we used 295 flowers way back. But now I continue this research, which I not incorporated because it's not published. After 10 years, I initiated another project. And now this is still going on. Now we scored nearly 10,000 flowers of the multiple population. The answer is the same. Honeybee is a zero efficient pollinator. She cannot do the pollination because of flower as well as the bee trait mismatching. Bumblebee, which we can see, the Bombus femoroidalis is considered as the pollinator because 83% of the flowers that receive the bumblebees are pollinated. And again, if you look at the club workers and the social groups in the bumblebees, unlike the honeybees, the queen of bumblebee also go for foraging, right? Honeybees, we all know that the queen will not be going out unless going for the nuptial flight, where she will be mating with the males and they will be producing, right? But in the case of bumblebee, you can see the queen at the beginning of the spring, you can see on the field, and she will be getting out the pollination. 100% of her visitor flowers pollinated workers, you can see the pollination efficiency is decreasing. Why so? Because bumblebee queen is still larger. She can touch even this anthostigma column in this depth, but worker cannot, because the worker, if you look at this picture, is short, right? A worker is smaller compared to this large queen, so she cannot touch as the time progresses. So now the question is, so that is the reason why there is an article came with the pollen.
Sorry to interrupt you, sir. Sir, actually, we are running out of time. So, kind.
Okay, I'll finish. Right, don't worry. So we already started late. So, but I don't mind. I'll be stopping at 3 sharp. Yeah. So that is what we find queen and worker bumblebees efficiency. So across the time, again, there is pollination efficiency of the worker bees is decreasing over period of time. This is 10 o'clock. It is so 11, 12. The flowers morphology is different. The bumblebee visited flowers, the fruit set is nearly 70%. Self-pollination is very less, suggesting that this plant is a cross-pollinated flowers. It is not only this, a large cardamom shows the such specialization. So the conclusion here is honeybee is a pollinator for small cardamom. For the large cardamom, it is a bumblebee is the pollinator. But specialization wise, the large cardamom is a specialized Electria cardamom is a generalist crop. But you look at this another gingiberacin plant growing in Nepal, you can see very long nectar tube for the flower, right? You can see the pollinator is nothing but a fly. There is no bee having such a long mouth part to access the nectar, because nectar foraging bee only or the fly can do the pollinator because of the flowers architecture, right? So with this, I'm not going to go for this cucurbitacin story. What I want to just finish with the slide that it is for plant pollinator studies is a very important aspect of research, and it is not that easy to identify the efficient pollinators in the system, and you should have both the collaborators from the plant scientist, or at least you should learn the techniques the plant scientists use to establish the pollination in a crop. Right, honeybees are not a universal solution, because many people will be keeping honeybee colonies nearby. In any of this amomum, I shown that honeybee is a zero efficient pollinator. So promote wild bees and other insects and manage habitat is very important for pollination, because the world is demanding now for wild pollinator conservation rather than depending on these honeybees. With this, I'm stopping. I'm a part, and in the university, we initiated a program called Indian Pollinator Initiative. And those who are interested can access the site in polling, not the site shown here, in polling.com, in polling.org. And scientists from Indian Institute of Science, ISOs, and many other universities are part of this. And if you want to be a part of that, you can access that. And if you're interested to go through this biology part of this, myself and Professor Shivana wrote a book on mutualistic interaction between flowering plants and animals. It is out of stock, I guess. Maybe the Manipal University Press will be you will be able to get a copy of it. With this, I thank the organizers for giving me an opportunity to share my findings. Thank you very much.
A heartfelt thank you to Dr. Sinu for illuminating our minds, which has opened new vistas in our understanding. We are now opening the floor for engaging question and answer session. If you have any queries, please feel free to share. Your thoughts are most welcome.
I'm sorry for rushing through because of time paucity, but you can also write to me if you have any questions. But I'm happy to take one.
I hope the response was satisfactory.
Hello.
Thank you, sir, for addressing the
Madam.
question.
Hello.
So,
Yeah. Am I audible?
you are.
Yes. Yeah. Sir, thank you very much. You have delivered a very wonderful talk on plant animal interaction, and the pollination is one of the important aspect. So I have one question. See, what plants we have discussed, those are mostly cultivated plants. Okay, like cardamom and those things we have discussed. So many times these are many pesticides are used for this. So pollinators they visit me because unless pollination is happened, we will not get the fruits. Okay, so does this pollination it affects or it has changed the
Yeah,
pesticide
I got the
changed the pollen?
question. Yeah, I got the question, Dr. Nilesh. See, finally, they are insects, right? So there are different perspectives actually. It all depends upon what is the detoxifying mechanism in insects, right? We have so many pests, and we are still trying to manage them with the pesticide, but the resurgence is happening for most of the pests. I really don't think that the bees are really affected by this exposure to the pesticides, but for a long term, right, they can have the mechanism to develop resistance in that, but that cannot be generalized answer for your question. It can vary. For instance, leaf cutter base, megachylid base don't have a very efficient detoxifying mechanism, so they are more susceptible to the contact pesticides, and compared to the honeybees. Honeybees have a very well established mechanism to detoxify the xenobiotics, right? So in that way, we cannot generalize this. You can say that this brinjal, every day you need a mug up of this pesticide treatment.
True.
Otherwise, the brinjal cannot grow. But
Yeah.
still, you can see a lot of pollinators roaming. But don't take it as a general statement. Research is showing that some cases bees are susceptible, some cases they are not susceptible. It all depends upon what is the detoxifying mechanism they have.
Okay. Yeah. Thank you very much, sir. Thank you.
Thank you, Suresh.
Thank you, sir, for addressing the question with such clarity and precision. Uh, I now invite J.M. Petare ma'am to express our sincere appreciation on behalf of all of us.
Thank
Over
you,
to
ma'am.
you.
Thank you, ma'am. Good afternoon, everyone. My heartfelt gratitude to Dr. Palleti Alessinu sir, our esteemed resource person, for guiding this insightful and enriching intellectual session today. You share knowledge about plant pollinator interaction for food security and how to manage pollination. Your expertise, vision, and thoughtful leadership truly made this session a memorable one. We are grateful for our valuable contribution to making this conference a success. Thank you very much, sir. Such a very informative session today. Thank you.
Thank you very much. Thank you. So I'm leaving. Okay.
Yes, sir. Yes, sir.
Thank you, ma'am, for expressing such collective
So,
appreciation with such warmth and grace. Before moving forward, I request all the participants to please switch on your camera and mute the mic for a minute to capture screenshot. Okay, thank you. Knowledge is light and learning is a path to progress. With such an enriching start, we now proceed to our technical sessions where eminent scholars will present their research findings and discussions will unfold around various themes of life sciences, biodiversity, and conservation. I now request R.P. Dadvi sir to conduct and regulate the session further. Presenters are informed to take your places as we begin the oral presentation session where research meets reality, where ideas turn into innovations. Best wishes to all the presenters. Over to you, sir.
Thank you, ma'am. Good afternoon to everyone. Now we will start the paper presentation. Before this, I will felicitate and introduce chairperson of this session, Dr. Milind Shinkade. He is working as an assistant professor and head of Department of Zoology in Sindhu Mahavidyalaya, Nagpur. His area of research, industrial entomology, pest control.
Excuse me, sir. Can you raise your volume?
His area of research, industrial entomology and animal tissue technique. Two research projects are completed under the guidance of Milind sir. And two students are awarded PhD and six students are working under the supervision of sir. Participant will present their findings and I request our esteemed panel of judges to clearly evaluate their contribution. Let us begin with our first presenter. Our first presenter, Dr. Parag Masra, who will speak on study of nesting behavior, adaptability in Asian weaver and Leucophilus smaragdina fabricius with respect to reproductive cycle and floral diversity.
ये बस पटकन बस पटकन चालू कर बस बस मी बोल परत रिपीट कर परत रिपीट कर सर सर केले स्टार्ट केले
Darvi sir.
Good afternoon, all. Shall I
Sorry
start
for
or
the delay.
I have to wait or shall I start?
Stay tuned and we will proceed further.
Good afternoon, Jutode sir. Shall I start? Wait.
1 minute, Parag sir.
Okay.
Dr. Milind sir.
Nilesh sir.
Yes.
Thank you very much, sir, for joining us. Shall I start the session, sir?
Yes, definitely. We can start with the first presentation. Presenter should know the 6 minute for presentation.
Yeah, okay.
So, Dr. Parag, please proceed with your presentation.
Thank you. Good afternoon, Milind sir. How are you?
Hello. Good afternoon. Well.
So first of all, I would like to thank Dr. Vilas Jutode sir who gave me an opportunity to present my research paper in this conference. The paper I'm going to present today is a part of my PhD work, which I've done. The title of the paper is Study of Nesting Behavior, Adaptability in the Ancient Weaver Ant, Leucophilus smaragdina fabricius, with respect to the reproductive cycle and the floral diversity of this ant. I can't change this.
Sir, we have given you the access. You can change it.
Okay. So basically, this ant, weaver ant, or the Asian weaver ant, it is an arboreal ant, and it is basically or normally found in the mango orchards And this ant has a presence, or you can say the distribution is cosmopolitan. It is found in central India as well as in coastal part of India, in the peninsular as well as in the Himalayan mountain ranges. This study was basically done to study the behavior of the ant in terms of nesting and selection of the host plant. So the parameters which were selected for this study was behavioral studies, the correlation of the construction of the ant along with the weather and the climatic condition, as well as this is the ant, it's a hymenopteran, it shows social behavior. The elite or the fertile caste of this ant uh, emerges only once a year. So the reproductive cycle with correlation to the nesting behavior was also studied. Material method, basically this was a behavioral study. So observations were done, data were collected from the nesting sites. Different nesting sites were studied, right from central India to the coastal part of Maharashtra. We studied different nesting sites. We come across different types of orchids. The results, first of all, I would like to show the kind of behavior this ant showed in the construction of their nest, where they constructed the nest by holding the leaf of the plant together with their jaws or the mandibles. And the workers attached themselves in a form of a live chain by holding each other's petiole or the appendages and pulling the leaves together. The and shows different types of workers. There are major workers who hold these leaves and pull them together. And there are minor workers who hold smaller first or second star larvae of the ant, which have a specific proper or a characteristic property or say a quality to secrete out a saliva, which when it comes in contact with the air, solidifies and form thin paper-like silky structure. So this thin paper-like silky structure is used by this ants to weave together this leaf. Weave means or to join together this leaf, and that's why the name weaver ant. It's kind of like they weave the leaf together to form the nest. So this is the tabular representation of the data what I have come across. If you look at the month or in the year, from month May to December, the construction of new nests were observed. They were happening, and the number of nests increased from month of May to the December. Not only the number, but also the height of the nest from the ground surface, as well as the population of the colony increased. Uh, is it visible? The size of the nest in centimeter? I see this table over here. Is the table visible to all?
Yes, sir. Yes, sir. It's visible.
So I'll just give you a diagrammatic representation how it happened. So from the month of May, in the month of May, the size of the nest was very small. With the from June to July and from August to September, October, and December, the size of the nest as well as the height of the nest from the ground surface increased. So if we look at in the month of May, we'll find a small cigar shaped nest, a pouch like nest, which was found around 5 feet, around 5 to 6, 7 feet of the from the ground surface. This basically carried all the fertile broods in them. The next was the month of July, August, a bit bigger oval shaped nest, which carried again the fertile brood. From September to October, the fertile brood emergence stopped completely because there was no fertile brood emergence from September to October. But the shape and the size of the nest increased, as well as the height of the nest from the ground surface also increased. And by the end of the year, like by November and December, October, November, December, the nest you will find always on the top of the canopy, which will be very bigger in size, and it will carry a huge colony in it. The diversity, or we can say the adaptability of this, and to select different types of plant for construction of the nest was observed over here. Basically, this ant prefers mango tree, but it was seen that this ant, or observed that this ant preferred any evergreen plant which is growing, which has leaf which are basically oblong in shape. Sometimes we also came across which is the family Astraceae, but they were not the primary selection or primary choice of the colony for construction of ant. The flora diversity comprised of mangifera indica or mango or guava or chiku. It was basically found, chiku plant were found in the coastal part of Maharashtra, where these ants were found to be selecting chiku plants for construction of nest. Then ficus as well as asoka and jamun. Jamun was not actually a favorable plant, but it could be possible that accidentally this ant could have constructed their nest. But if the surrounding does not have that many of the mango plants, these ants can adapt to any different types of plant, but only condition they has to be evergreen plant. So this ant show a great adaptive quality in terms of selection of the host plant as well. These are certain pictures which I have clicked. This is a chiku plant. You can see this nest was a kind of used constructed using two different branches. It is kind of a
Excuse me,
nest.
sir.
Yeah, my time is over.
Actually, it is a kind reminder that you have a limited time of
Okay.
just 6 minutes.
Okay, so this is chiku, mango, guava plant over here. This is a jamun plant, again a chiku plant. Uh, just observe the shape of that. Another thing which I came across was the direction of the nest, in which the nest have been constructed. They were always in the northeast direction. The reason for this was observed because the southwest monsoon which comes to the peninsular part of India, the nests were always constructed opposite side of the southwest monsoon. The fourth category of nest were during the October, November, December. By that time, the monsoon is over. So the ants can also adapt themselves as per the climatic condition. This were the data, the tabular data for which the last week of May to the mid week of July. And you can see as the height and the numbers of leaf increases, the numbers of the fertile wood also increased. So by the mid of July, emergence of fertile wood was on the full flesh flow. By last week of May, in the central part of India, as well as the western part, basically western part of Maharashtra, the fertile wood starts emerging. Right now, if the monsoon occurs early in the peninsular part, we also get the occurrence now. I think we are getting occurrence of the fertile brood in the state of Kerala. So where monsoon occurs early, the occurrence or the emergence of this fertile brood also comes early. To discuss it, that this ant use themselves as a life chain, just like how we use bamboo or other material to construct their own nest. They show a great variety of adaptability in terms of selecting their host plant as well as adapt themselves to the changing climatic condition. They change the shape of their nest as per the season as well as the height of the nest from the ground to adapt or to overcome the climatic condition or the variation in the temperature. The lower the nest near the ground surface, the cooler it is. Wherever it goes up, it becomes a bit hotter. The characteristic or the quality shown by this ant in constructing the nest against the southwest monsoon wind, the opposite direction of the southeast monsoon wind also proves that these ants also show in a kind of a quality or an adaptability to the environmental change. So there is one paper I've done with the help of we have done ultrastructure where the sensory organs of this ant can have a quality of judging the change in the atmospheric temperature as well as moisture. So the conclusion, as I said, the ant shows a great deal of adaptability in terms of climate as well as the nesting habit. Also, it changes its own reproductive cycle. As I said, in the state of Kerala, we can find out the emergence of brood has started, but in Maharashtra, we don't come across until the monsoon occurs. So this was the paper. This is the conclusion. These are the references which I have used. This is the paper which I have published already on this title. Thank you.
Okay, nice presentation, Dr. Parag, on the weaver ant, Asian weaver ant.
Thank you, sir.
Any question from participant? No, I would like to ask one query when we are talking about the weaver ant or arboreal ant specifically. Most of the time, the ants which are forming the nest on the land, they are also having the opening from east side. So basically, when we are talking about the weaver ant or arboreal ant, there is a relation of behavior with the humidity, as you said.
Yes, sir.
That means humidity.
Yeah.
So basically, in our region, in Nagpur region, badam plants are there where, or Pongamia pinnata, karanj plants are there where, yeah, these ants are specifically found there, generally have their nest. So any other typical behavior of this ant specifically in case where you are saying that the leaf is a typical leaf, oblong shaped leaf or round shaped leaf? Any other specific behavior towards that plant particular, because they are choosing this plant specifically?
Sir, what I've come across during the study, I've seen that all those plants which had oblong leaf, if they're available near the colony, they can form this type of cigar-shaped nest to have these roots. And that too, right from the end of month of May to the first or second week of June, till the monsoon occurs. But I've also come across one thing in my own college campus, where the ants were given a plastic bottle. I kept a plastic bottle covered in a normal newspaper, and the ants started forming nests in that also. So it is it is a topic of research why these ants choose such shape to form the nest only in that period for brooding purpose, and after that they move upwards in the canopy and they form a oval shape of nest.
Okay, so basically canopy is important. Either they are made up of the leaves or anything like bottle or paper.
The height of the nest from the ground, as well as the plant should be an evergreen plant. If the plant is a deciduous plant and sheds its leaf during the summer season, then the colony will disturb. So that's why they choose mango and other evergreen plants. And the shape of the leaf is the secondary thing. The first thing has to be an evergreen plant.
Okay. Good work.
Thank you, sir.
Yes, next, please. Any question? No.
Okay, thank you.
Okay, thank you.
Thank you, sir. The next presenter name is the Pangvi Ramakrishnan, who will speak on Beyond the Bloom, the ecological role of extrafloral nectaries, and it is a sweet seed. Unlocking the secret of extrafloral nectaries in plant protection. I request to Pangvi Ramakrishnan, please present our topic.
Am I audible, sir?
Yes,
is my presentation is visible, sir?
Yes, ma'am.
Good evening to all present here. Myself, Dr. Pangvi Ramakrishnan, working as assistant professor at Joy University in School of Agriculture Science at Tirunelveli. I'm going to present a review topic Beyond the Bloom, the ecological role of extrafloral nectaries in pest management. This is one of the, this is my whole PhD research work. I'm going to throw some light on this. We all know about floral nectaries. Many have not unaware of this extrafloral nectaries. These nectaries are the specially structure present on the mostly on vegetative part and lessly on the reproductive part of the plant. They are not directly involved in pollination, but these nectaries will act as an indirect defense mechanism against herbivore. Like this plant will produce some nectar. When the plant is affected by any herbivore, it will increase the production of nectar, thereby attracting the natural image to the particular plant and acting as a food source, thereby the indirect defense mechanism will be taking place. First image show the extrafloral nectaris, where the nectar can be secreted there, and the second image is extrafloral nectaris present on the castor stem, and these are the extrafloral nectaris present on the leaf, and this is the extrafloral nectaris on the calyx, that is reproductive part of hibiscus cannabinus, that is mesca. Coming to distribution of EFN bearing plants, EFN, these nectar structures are present in 3,941 plant species belonging to 745 genera with 108 families. These are the list of the plant families like Fabaceae. In Fabaceae, cowpea, prominently we can see the nectar structure. In Euphorbia and castor, we can see the prominently we can be able to see Convolvulaceae like Hypomecarnia, Rosaceae, Anthraceae, and Indian almond. And the Calaceae, Cucurbitaceae also you can see structures like EFN structure where the ants get mostly attracted, and Malvaceae and Pedalaceae. In Malvaceae, I have worked on hibiscus cannabinus and Subberifa plant during the PhD research work. And these structures are present on the spikes, pedicel, buds, calyx, and that is all over the part of the plant except on the roots. And the first image shows the EFN structure present on the leaf of hibiscus cannabinus. This is the structure. And the second image, EFN structure present on the reproductive part. And the third one is EFN structure present on the hibiscus subberifa. This is the structure where the nectar is produced. And the down part is the nectar structure present on the suborbal plant. This is the micromorphology structure of extrafloral nectaris. The structure may vary in different shape. It may be round, it may be thick, it may be a narrow shape, it may be wider, it may be round in shape. In case of hibiscus cannabinus, it is round in shape. In case of hibiscus subberifa, the slit is nectar narrow in structure where the nectar is secreted. Coming to gland activity pattern, so very less research was done in the extrafloral nectar secretion in season wise. And during my research, I also concluded that the dry season has required more number of extrafloral nectar secretion. That too, temperature plays a very important role when comparing to other factors. And number of EFN present on the plant will also influence nectar production. Age of extrafloral nectari, where nectary structures are surrounded by some secreted trichomes. These trichomes will also produce nectars, which act as indirect defense mechanism against herbivore. I have come across articles last third image that the glandular trichome has defense against the herbivore producing some nectar. And this is the first image where you can see the nectar structure with the nectar production on the plants of hibiscus cannabinus and Subberifa. The nectar is like honeydew droplets. Coming to impact of EFN on extrafloral nectaris on measurements, many articles have come across. Two treatments have been imposed, one with EFN present plant and another was EFN absent plant. For example, if in case of EFN absent plant, the plant structure, that is EFN structure is masked by using the nail polish, so that we can prevent the production of nectar that can be done weekly interval and population of herbivore and predators and parasites are prevented. It has resulted that more number of natural enemies like predators and parasites have been recorded in case of EFN bearing plant when compared to EFN present plant. I have given some example. EFN contribute to reduction in Bacillus molesta egg, larva, and pupa in peach orchards. And Phaseolus liliotines, that is lemma beans, will attract more number of ants, wasps, and flies when compared to non-EFN plants. And within the plant species also, we can see the difference of attraction of herbivore as well as naturalness towards the plants. And Solanum adherens having extrafloral nectar structure, but it is absent in case of Rudbeckia. The Solanum adherens attracted more number of parasites and more parasitism observed in case of Solanum adherens species when comparing to Solanum luteum without EFN plants. So we can see the first image, we can see the red cotton bug, and the down part, the second image showing the later, and the rest of the two images where the mealybugs were parasitized by Anaceus arizonensis parasite. These are recorded in the EFN bearing plants of Hibiscus cannabinus, that is commonly called as Mesta. And some of the predators also feeding on EFN bearing plants. The first one is coccinellic grub feeding on the mealybug, and the second is Physophila eggs are laid in between the Phenacoccus solanatus, that is mealybug of Mesta, where the third image is the grub of the Physophila is effectively feeding and mimicking as a mealybug and feeding on the mealybug of the Mesta and reducing the herbivore population. So it influences on the yield. Nalini and Revati is one of my PhD guide. She has worked on this EFN. She also reported that EFN has increased the more number of infestation, not only the ants but also more number of predators and parasites in pasture cotton and zazana, as well as Matthews and his co-authors in 2009 has reported more number of predator population and parasite population were reported in case of EFN bearing plants and increase the yield. And when compared to without EFN. So this extrapole bearing crops have recorded more number of population when comparing to non-EFN bearing plants, and more pollinated population that also has been recorded. During the research work, more number of pollination, even though the Hibiscus cannabinus is self-pollinated crop, now recently now cross-pollination has been reported in case of Hibiscus cannabinus in Sub-America, maybe due to presence of this nectary attracting more number of pollinators to the plant. So the cross-pollination has been happening in case of Hibiscus cannabinus in Sub-America. So apart from that, this EFN has been tested for lab studies of particular parasites, which increase the survival of predators, not only predators as well as parasites. I have also checked the parasite like Anaceus arizonensis and Albina, where increase the longevity, survival, fecundity, and sex ratio of the female with regard to parasite feed on EFN nectar and comparing to honey as well as the control. So coming to conclusion, this is EFN bearing crops as one of the including the potential of predators and parasites which can be used as food source at the laboratory level, and these EFN bearing crops can be added as a component in integrated pest management, and this is one of the zero cost conversion method of natural use where this is today we are using more number of chemicals and polluting the environment and affecting all the minute organisms. So we can add these kind of crops in IPM pest management so that we can achieve the pest population easily. And in near future, we can also extract this EFN structure, EFN nectar from the plant, and through which the pheromones can be synthesized for
Sorry to interrupt you, ma'am.
to attract the natural enemies.
But
Okay,
keep in mind that you have a time limit.
Okay, this is the last one. So here my conclusion, the world of diminishing resources. It's time to work with nature and shift our focus and simply understand the insect symbiosis to harnessing their potential to provide effective and low-cost pest control options. Thank you. Thank you.
Okay, thank you, ma'am. Good presentation on your topic, which is ecological role of extra floral nectaries.
Floral nectaries.
Any question from the participant? Okay, I have one query. Have you gone through any specific methodologies of the means extra floral nectaries that generally defended by this pest? As you said, the ants are.
Yeah, I have collected during the PhD research. I have collected the nectar by using the microcapillary tubes, and I have tested for the parasites in the laboratory level, especially for mealybug parasites. One is Anaceus arizonensis and Anaceus albina. which I've seen in the which are shown in the slide where the mealybugs are attacked by the many of the parasites, small brown color. That is the parasites I have collected. I have given as
Okay.
a food source in laboratory level. They, in comparing to honey as well as in sucrose and fructose, it increases the survival and longevity. I have collected the nectar from this plant. I have shown the information that I have collected. Practically,
Okay.
I have
And
done.
nowadays, these ENFs are used as a pheromones. Means these kind of regularities are now there in
No,
some. It can be used.
now they have, it can be used. It is my future conclusion.
Okay.
Now only these kind of crops and pest management we are taking by most of the research are done in the abroad countries only. In India, only less amount of work is done with this with regard to pest management. So you have to
Okay.
work on this to reduce the impact on environment.
We have the plantation of that kind of species for the extra floral nectars, basically.
Okay, sir. Okay,
Yeah.
okay, sir. Okay, sir.
Okay. Thank you.
Thank you, sir.
Thank you, ma'am. The next presenter name is Dr. Digvijay Kumbar, sir, who will speak on qualitative analysis of vermicompost produced from two different methods employing earthworm, Isnea foetida. Sir, I request to present our topic, sir.
Sorry to interrupt you, sir. There is an announcement. I wanna inform you that the parallel session of oral presentations is ongoing. So I kindly request Ms. Hemal Dhage and Mr. Daulat Wagmore to leave the meeting and join the separate link of Google Meet that has already been shared with you. Please ensure that you are connected to the right meeting. Thank you. Sir, you can go ahead.
Is the screen visible?
Yes, sir.
Okay. Okay, so I'm Dr. Digvijay Kumbar, and the topic of my presentation is qualitative analysis of vermicompost produced from two different methods employing earthworm species, Icenia foetida. So the vermicompost is considered as one of the organic fertilizer and acting as a soil conditioner. And vermicompost is used for conversion of soil waste into nutrient-rich material. So importance of vermicompost as a plant growth promoter that increases soil fertility, and vermicomposting technology has paved a way for effective organic waste management. So the behind the two objectives, basically we have prepared behind the study, that is to study the best raw material for the vermicomposting and to compare the quality of vermicomposting produced from the two methods by analyzing the physical properties of it. So as far as the material and methods are concerned, the study area of vermicompost is our college itself, vermicomposting unit. And the material we have used is the garden waste, cow dung, rice straw, paper waste, earthworm, and that is Icenia foetida, and HDPE vermicompost bag. So the paper waste and garden waste were collected from the Dalapati Mahavidar campus itself, and cow dung was collected from the nearest village of the Nandgaon, here in the near to Karjat. The rice straw was collected from the agricultural field situated in Kolhapur, and to use authenticated species of earthworm, Icenia foetida were collected from the vermicompost unit of Dalapati Mahavidar Karjat, and HDPE vermicomposting bag was purchased from online, so that is Amazon. The preparation of vermicompost bed in method 1, we have added thin layer of earthworm culture at the bottom of the tank, and then we have prepared a layer of compost of rice straw that is 5 to 6 inch. Again, that this layer was followed by the paper waste, again rice straw, and again paper waste. That is 2 layers of rice straw and 2 layers of paper waste were formed. In method 2, firstly we have the layer of the earthworm culture at the bottom of the tank, and the layer composed material garden waste, that is the 5 to 6 inch, and then cow dung, then garden waste, and then cow dung. There is 2 layers of garden waste and 2 layers of cow dung were incorporated into the vermicompost bag. The parameters we have analyzed, they are the moisture, total organic carbon, TOC, pH, electrical conductivity, that is EC, total nitrogen, total phosphorus, total potassium, total sulfates, iron, then manganese, zinc, and copper. So there are total 12 parameters we have analyzed from the vermicompost obtained from the two different methods, and these are the different methodology or techniques we have used or incorporated for the analysis. So this is the observation while at the middle when the earthworm starts decomposing the paper waste. As far as the results and discussion are concerned, the moisture, the percentage is in method 1, that is 38.6, while in method 2, 54.2. Total organic carbon 26.16 in method 1, 30.60 in method 2. pH is 6.95, that is very near to the neutral pH, and 6.63 in method 2. EC in method 1 3.40 and recorded as 3.49 in method 2. Total nitrogen 1.48 in method 1, 2.87 in method 2. Total phosphorus 0.78 in method 1 and 2.13 in method 2. Then total potassium 1.20 in method 1, 1.34 in method 2. Then total sulfates 0.38 and 0.61. Iron 151.75 ppm and 201.21 ppm. Manganese 98.64 and 131.24 ppm. Zinc 72.606 and 92.41 ppm. Copper 21.53 and 28.29 ppm. So these are some graphical presentations of the results obtained. In method 1, the total organic carbon is as compared to method 2 is less, that is in method 2 30.86. Then moisture content, as far as the percentage is concerned, the method 2 is having ability to hold the moisture more as compared to the method vermicompost produced from method 1. And then iron, manganese, zinc, and copper. All the parameters or the physiochemical parameters shows increased value or little bit increased value as compared to the method 1. EC is also having great impact with the method 2 as compared to method 1. And nitrogen, phosphorus, potassium, and sulfate, that is, these are the main compounds which are generally required for the growth of a plant. They are maximum in method 2 and as compared to the method 1. So the study revealed that the physiochemical properties of vermicompost can be prepared by using two different methods have good values as compared to any other organic compost. That is method 2, that is cow dung and garden waste, indicates higher values of all the physiochemical parameters as compared to method 1, rice bran paper waste. So the substrates used influence physiochemical and biological properties of vermicompost and are recommended for vermicomposting and its production. So these are the references. Thank you.
Okay.
Thank you.
Yeah, nice presentation. Any question from participant? Sir, any other species we can use for this particular vermicomposting method other than Icena foetida?
Yes, sir. One more species generally used in India, that is the Uterus eugenie, red worms. These are the red regulars. But as far as the reproduction rate is concerned, the Icena foetida is very well equipped or very well adapted to this rate is also high.
Okay. Okay. Okay, sir.
Okay,
nice presentation.
Thank you very much, Dr. Kumar, sir.
Thank you. Thank you. Thank you.
Okay, next presenter is Dr. Monali Kadu. Start the session, start the presentation.
Am I audible? Hello. Sir, am I audible?
Yes, ma'am.
Yes, audible.
Yes. Good afternoon, one and all. Myself, Monali Kadu, working as a PhD student under the guidance of Dr. Ritesh Kumar Chaudhary at Agarkar Research Institute. So today I'll be presenting a part of my objective. As we all know that medicinal plants have various therapeutic value, but however, they are misidentified, and also there are various adulteration problem assigned with it. That's why pharmacognosy and standardization of medicinal plant is very much important. So let us begin. I'm working on plant haplanthode, which belongs to family Acanthaceae. There are 5 taxa in haplanthodes, which are listed over here. Haplanthodes are endemic to India, and mostly it is distributed to the western parts of India. So according to the recent study by Surveshwaram and his co-workers, they have done molecular phylogenetic studies. Here they found that Andrographis and haplanthodes is closely related to each other. And Andrographis is also commonly known as calmate. It has been tremendously explored for its medicinal value. That is why we hypothesize that the similar or novel compounds may be present in haplanthodes also, which may cure various diseases. We identified the research gap that the standardization data was unavailable for haplanthodes. That is why we aimed to confirm the identity of haplanthodes. We were curious to understand the key questions. Could the haplanthode standardization data, the phytochemical analysis and antioxidant analysis help to ensure the quality in pharmacognostic research? We also found some of the ethnobotanical leads. Most of the tribal people used haplanthode as a febrifuge for boosting immunity, etc. For understanding the locality of haplanthode, we visited various libraries and also consulted online as well as offline herbarium. After which we acquired the permission from the Biodiversity Board, and then we collected the plants from the western parts of India. Here are some of the collection photos while collecting haplanthodes, and here is the state-wise location which we covered in search of haplanthodes. We identified haplanthode by using the key characteristics, which is shown in this chart. After which we preserved the specimen by preparing the herbarium of each samples, and then we maintained the excision for all the 5 species. Here is the methodology which is given in the chart form. Here, after understanding the ethnomotical leads of haplanthode, we collected the plants, we dried them, we grinded them into powder, then we cold macerated using methanol for 72 hours, and then we used various polarity solvents, and then we rotovapped them to obtain a condensed plant extract. And this condensed plant extract was used for pharmacognostic evaluation and phytochemical analysis, which I will be discussing further. So here is the photoplate for powder microscopic study for haplanthodes, all the 5 species. Here we found the key characteristic that cistulids was present in haplanthodes, all the species. After which we did the anatomical study to each haplanthodes. We also did the stomatal study in which we found that diacetic stomata was present dominant in all the species. We carried out standardization. One of the parameter is ash value analysis. Here haplanthode nilgiriensis leaf showed the highest ash value. After doing the extractive value analysis using various solvents, we found out that water had a highest extractive value in haplanthodes. We also performed the qualitative and quantitative phytochemical analysis, and here abundantly we found the phenols, flavonoids, and tannins in haplanthodes. That is why we carried out quantitative analysis by using the spectrophotometry method. Here is the procedure for total phenolic content, flavonoid, and tannin content. We found out that the maximum total phenolic content was found in haplanthode verticillata, ethyl acetate extractive. The maximum flavonoid content was found in haplanthode plumosa N-hexane extract, and the maximum tannin content was found in N-butanol extract of haplanthode plumosa. Thus, here we have discussed a significant number of pharmacognostic analysis, like the total ash value, extractive value, the microscopic analysis, etc. This standardization data will help to augment the supply of authentic crude drug And thus, this will provide the insight for many pharmacological companies in drug development. Our future focus will be on the second and third objective, that is the metabolite profiling, and also the assessment of pharmacological activities of haploids. Thank you.
Okay, madam, nice presentation. Any question from participant?
Thank you, sir.
Okay.
Next presenter name is Dr. Deepak Kadam. Sir, please start our session.
Start yours.
Am I audible?
Yes, sir. Yes.
Just a minute. Hello everyone, I am Deepak Kadam, presenting my research work entitled as Assessing Disease-Associated Non-Synonymous SNPs in Human Catalase using Integrated Bioinformatics Approaches. I want to mention that my research will fall under the one of the themes provided by conference organizers, that is molecular biology and genetics. Persistent hyperglycemia induces ROS production, reactive oxygen species production, which, if not scavenged, damages all cell molecules. ROS damage DNA and RNA by breaking phosphodiester bonds, which stops cell division and apoptosis occurs. In case of cell membrane, excessive ROS forms lipid peroxidase as membrane is rich in lipids. It also carbonates proteins, which leads to protein aggregation and reduces degradation of non-functional proteins. To cope up with ROS, cells have developed antioxidant response, which include antioxidant molecules, glutathione, thioredoxin, peroxidine, uric acid, and bilirubin. Antioxidant enzymes include SOD, catalase, GPX, GR, H1, etc. As we all know that catalase is an antioxidant enzyme that breaks down hydrogen peroxide into water and oxygen, protecting cells from oxidative damage. The single nucleotide polymorphism in promoter region of antioxidant enzyme genes can affect their expression. This can compromise individuals' antioxidant response. SNPs in most important antioxidant genes such as SOD and catalase have been well recorded. However, reports of SNPs within their promoter region are very few and are not many reports, especially in Indian population. So the objective of the study were to locate the SNPs in the promoter region of catalase and check their association with catalase enzyme activity in controlled non-diabetic and diabetic individuals in Indian population, and to check the functional relevance of the SNPs in the promoter region using reporter assay. Because of time limit, I will not focus on material methods. I will focus on the results only. Samples were collected. In samples, fasting blood sample were collected from 19 non-diabetic and 98 diabetic individuals. The samples were prepared by centrifuging them to separate plasma and erythrocyte fractions. Catalase activity in serum sample was determined following standard protocol. In results, you can see here, diabetic individuals had significantly high concentration of fasting blood sugar and HbA1c. Catalase activity was significantly low in diabetic individuals than in control non-diabetic individuals, indicating oxidative stress in diabetic individuals. DNA were extracted, genomic DNA was extracted from whole blood and checked on 1% agarose gel electrophoresis. To amplify the promoter of catalase gene, primers were designed which would cover the entire core promoter as shown in the figure. Then the catalase promoter amplification was done by using standard PCR reaction mixture. As you can see here, the PCR products were purified and the purified amplicons were sequenced using Sanger sequencing at NCMR NCCS Pune. In catalase sequence analysis, we detected 6 SNPs reported in DBSNP in catalase promoter region, of which only 3 were found to be polymorphic among our studied groups. These 3 SNPs were found to overlap with enhancer, which 238 different transcription factors can potentially bind. In both control and non-diabetic, non-diabetic and diabetic groups, similar transcription factors were found to bind at minus 89 and minus 20 positions. However, at position 330, minus 330, instead of GATA1, serial transcription factor was found to binding at the diabetic group. However, GATA1 and serial have not been reported to activate CAT transcription. Further, to check the SNP functional relevance in catalase gene promoter, reporter assay was performed. In this, luciferase gene was placed in front of the both the wild type and promoter containing SNP for catalase gene, and activity of luciferase was measured. A significant difference was not observed between luciferase activity from wild type or SNP containing promoter of catalase in reporter assay. This indicates that these SNPs do not significantly affect the catalase expression. In conclusion, CAT promoter region were observed 6 reported SNPs in the 921 base pair sequences, out of which 3 were polymorphic in control non-diabetic and diabetic groups. Further, allele T of 1179 was found to be significantly associated with the disease catalase activity in diabetic individuals. This is the first study reporting 3 polymorphic catalase SNPs in Indian diabetic population. However, there was no association was found between these polymorphism and reducing antioxidant activity in diabetic individuals. Thank you.
Okay, sir, this is a very good work, novel work. As you said, the 3 SNPs are only working for non-diabetic and diabetic populations.
Yes.
Okay. What is the role of luciferase basically when you are using the
means in which if the luciferase express, it will emit light or a signal, and we can know that our SNP is working or not. If it is not working, the signal will not come out.
Okay. Any other radioactive compound or tag we are using other than luciferase? Because luciferase enzyme is not as well illuminating every time, I think.
Yeah, you are right, sir. Actually, I have done only one reporter assay till now. So we have used this as referenced in other citations also. So
okay, no problem. Good work.
Thank
All the best.
Thank you, sir. The next presenter name is Miss Triloki Zaveri. Ma'am, please present your topic.
Hello, hello.
Yes, sir.
We have to finish before the time. So give to 3 minutes only. Hello, just a minute. So we have to finish within 3 minutes. Just go to the result and conclusion.
Okay, sir. Good afternoon, everyone. Today, my topic for presentation is pharmacognosy of traditionally important flask amalthas. The aim was to standardize the standardization of the flowers of Acacia fistula. And its main reason for standardization was because the decoction of the flowers are used for treating many skin infections and wounds and fever infections also. The classification states that it belongs to the family Fabaceae, subfamily Papilionaceae, commonly called as botanically called as Acacia fistula and commonly called as Amalthus. For standardization, different parameters are being taken care of. For further standardization to take place, the first thing is collection of the plant sample and then the authentication of plant sample, which was done from St. Xavier's College, Mumbai, and the further processing was taken care of. The next is organoleptic and microscopic characters, where for a flower, there are four basic moles present: calyx, corolla, androecium, and gynoecium. The calyx has 5 petals, polypetalis with velvet aestivation and green and smooth on both the surfaces, whereas corolla is again 5 petals, polypetalis alternate to calyx, having smooth inner surface and hairs on the outer surfaces. Androecium are 10 in number with 7 smaller ones and 3 completely matured long anthers with only trichomes having on the anthers. Gynoecium is basically a single stalk having a thin stalk at the base, then the whole ovule and a single stigma with a white tip. For the microscopic sections, the TS of calyx and corolla both was taken, having a single layered epidermal cell with a thick cuticle in both calyx and corolla, followed by parenchymatous cells, vascular bundles, lower epidermis, and trichomes on both the surfaces. The major thing was noticed that the stomata are present at an intervals in corolla. The sections of gynoecium and anthers were also taken. Next is histochemical analysis. The histochemical analysis stated the sections treated with different chemicals to check the presence of tannins, amino acids, proteins, alkaloids, and also some calcium oxalate crystals were found in corolla. The powder analysis showed the presence of petted vessels, epidermal cells, and collenchyma cells in calyx, whereas in corolla, different types of trichomes was seen. Then also prismatic crystals and calcium oxalate crystals and petted vessels. The major difference that was seen was there were 3 to 4 types of trichomes seen in the entire powder, which were glandular trichomes, simple trichomes, bicellular trichomes, unicellular trichomes, even oil globule stomata were also present. The preliminary phytochemical screening was further taken, in which proteins and amino acids were present in all the 3 samples. Whereas flavonoids was present in very high concentration in all the 3 samples, especially in the methanolic extract. If you go ahead with physicochemical analysis, you can
Madam,
see
please
that.
go to result. Madam, please go to result. Conclude the session. Please go to result.
Sir, it's just 1 minute. Okay. The pharmacognostic test was taken and the heavy metal test was also further done. The conclusion is the pharmacognosy is done for the first time for the flowers, and the unique characters will help in distinguishing it from one flower to another. And these standards will help in the authentication of the drugs. The further plan is for the quantitative phytochemical studies and pharmacognostic studies will be carried out. These are my references. Thank you.
Okay, nice presentation.
Thank you, sir.
Thank you, ma'am. The next presenter name is Dr. Manoj Thakre. Sir, please present your topic. Dr. Manoj Thakre, sir. Sir, please present your topic. Sir, please start your presentation.
Can I audible, sir?
Yes, sir. Yes, sir.
I am audible, sir?
Yes.
Okay. Okay. My name is Manoj Thakre from Department of Zoology, RTM, Nagpur University, Nagpur. And the topic of the presentation is conservation and biology of tropical tussar silkworm Anthraemylata, Ecorus bandara, Lepidoptera saturninii. Next. There are 4 different kind of the silkworm cultivated in the India. They are mulberry, tussar, muga, and eri. They are cultivated at different ecological pyramids of the India. Basically, the tropical tussar silkworm, which is called as the Anthramalata, generally this race is cultivated in our tropical belt of the India. Means next, it is generally found in our region. So in the introduction part, 4 different kinds of the variety cultivated in the India. And in India, Maharashtra is the traditional tussar producing state. And in Vidarbha, Bhandara, Gondia, and Gadchiroli districts are known as a traditional tussar silk production districts. There are different 44 ecoregions which is found in the India, and they are on the basis of the ecological niches as well as the quality of the cocoon and the cocoon production. Next. Then the geographical distribution of this Anthrea malata ecoris Bhandara, this race is actually found in the Vidarbha region, the East Vidarbha region, that is Bhandara, Chandrapur, Gadchiroli region and Gondia region. The ecorace, the homeland of the ecorace is the Guikhuri ranges, which is generally extreme from south to west corner and the tracts of that Bhandara region. Vidarbha region, in Vidarbha region, it is also called as the Mulki or Ranat in the adjoining areas of the Madhya Pradesh and the Chhattisgarh. Next. This is actually the geographical distribution, which is you see in the east Vidarbha region, and the in Vidarbha region, the 4 districts are actually important for the cultivation. Next. Now, this race is now at the brink of extinction. Why? It is because there are so many different reasons. Why this race is to be the conserved. So the conservation, it is because it is very much economic important of its cocoon shell, cocoon weight, higher silk ratio, higher denier silk, and the disease resistance capacity, which is not seen in the another races which is generally cultivated in India. But due to the unchecked exploitation and collection at the tribal community and directly they sold into the market, the grainage and the seed preparation is not there. And hence, it is very necessary that we have to conserve this ecorace, precise ecorace, because it is indigenous. Next. Now, these are the comparison, the 3 different ecoraces of Anthera malata, which is actually cultivated for the commercial purpose, that is the Daba, Bhandara, and Sukinda. So, when you compare Bhandara ecorace with the Daba and Sukinda, it is always a stand first. Okay. And because of it having some physical and economical characteristics, is somehow less than the Daba, but it having a very important property, that is in case of the bacterial and viral infection. Because it is indigenous, so naturally the bacterial, viral and protozoal infection is seen very minimum, and hence it having a high cocoon production and the high silk content. Next. This is classification. Next. Life cycle, which consisting the stages, egg, larva, and the third cycle. Now this is the duration. Next. This is the duration, larval duration, emergence, and oviposition. So duration is also in the third crop. Always we consider the third crop. This is multivoltine race. So third crop is always benefited, always economic. So larval duration, emergence, and oviposition, embryonic stages, and spinning pan of this Bhandara ecorace is always stand higher. Next. Now, what is the conservation strategy? How we conserve? How we develop the strategy? The conservation is thus not simple, because directly you in situ and ex situ conservation are there. But when you go to the field, then there are different collective measures which is taken into the consideration. So participation of local tribal people for the conservation strategy, collection of the cocoon from tribals. Then with the help of tribal, we have to
Sorry to interrupt you, sir.
Yes, madam.
Actually, we are running short of time, so kindly
Okay,
conclude,
give me only 2 minutes. I conclude 2 minutes. Okay. So rearing in situ and the field rearing and awareness among the people. Next. So these are some strategy, collection of the cocoon from tribal people. Then grainage from the Department of Zoology, RTM Nagpur University, as well as the field rearing. Next. Then this is the field training centers. We develop 4 training centers as 4 different districts. And then again program at different what you said, the centers. And last one, that is the involvement of the women in conservation. This is very important, because women are very much part of our main focus on the women, because they actually participate in this our program. And now we've seen the conservation or the multiplication of this race is seen. Next. Conclusion: with the help of tribal and nomadic people, this eco-research Bandara cocoon collected, and then multiplication is seen. Multiplication is done with the in situ and ex situ rearing. Then the awareness among the people, entire cocoon population from the forest, and only with the help of local tribal community, this conservation is possible. So these are the references, and this is the acknowledgement. We are thankful to RGSTC, Government of Maharashtra, Mumbai, as well as Director of Sericulture, Government of Maharashtra, for their timely help. Thank you so much. Thank you, sir. Thank you, ma'am.
Thank you, sir, for good presentation.
Thank you, sir.
The participants showcased commendable enthusiasm and excellence through their presentations. Heartiest congratulations to all of you for insightful contributions. It's now my honor to invite our esteemed principal, Dr. M.T. Sarode sir, to deliver the welcome address for the valedictory function. Over to you, sir.
Second parallel session has finished or is going on?
Sir, ongoing.
Sir, it is still running.
Few words about the today's resource person, Melin sir. 2 minutes you conclude these presentations.
Okay, sir.
Milin sir.
Yes. So I congratulate all the team for this conference, Advances in Life Sciences, Biodiversity and Conservation. So this is a session for online oral presentation where the 7 or 8 participants have presented their research work. And basically it is all belonging to the new concept or new era presentation for the conservation and application aspects of the various entomological insects or things like pharmacological or pharmacognosal importance in the region of agriculture and pharmacy field. So I congratulate to the team and college for this taking efforts for good conference and giving chance and opportunities to the various researcher in their upcoming aspects of the applications and economic importance of the insect. Basically, when nowadays we are going to implement and regulate the various aspects of the Indian knowledge system, indigenous knowledge system, as well as new education policy. This is important in the showcasing the zoology or a life sciences, particularly in the mode of this particular aspect, where the various animals or the diversity, either it is a plant or any other bacteria or novel concepts of the engineering and biotic engineering. So these aspects are very good to present in the respect of national education policy, because it was implemented 1 or 2 years back, but nowadays the skill and opportunities for this life sciences is very important for in this particular regard. So I again congratulate all the researchers, specifically from near in the Paschim Maharashtra or in Vidarbha. All researchers are stick to their particular perspectives of the applications in insects and entomology. So very good presentations in this particular session. I am thankful to Principal Sir as well as Dr. Vilas Jutode giving me this opportunity for as a chairperson for this particular session. Thank you. Thank you so much, sir.
Thank you, sir.
Thank you, sir. Now I want to extend a note of gratitude to our chairperson, Milin sir, for your great contribution and chairpersoning the session. Thank you, sir. Now we are starting with the valedictory function, and it is my honor to invite our esteemed principal, Dr. M.T. Sarode, sir, to deliver the welcome address for the valedictory function. Over to you, sir.
Okay, thank you, madam. Hilal, sir. Hello. Hello.
Hello.
So we started a valedictory function.
Yes, sir.
Okay, thank you. Good evening to one and all present here. It is my great honor and privilege to extend a warm welcome to all of you on the occasion of the valedictory function of the International E-Conference on Advances of Life Science, Biodiversity and Conservation. Over the day, we have witnessed an enriching exchange of knowledge, insights, and experiences from distinguished scientists, researchers, academicians, and students from across the globe. This conference has served as variant platform to discuss pressing issues, innovative solutions, and future directions in the field of science and life sciences and biodiversity conservation. On behalf of the Sri Sadguru Gangagir Maharaj Science, Gautam Arts, and Sanjeevani Commerce College, I take this opportunity to welcome our today's chief guest on this occasion, Dr. Honorable चंद्रकांत खिलारे साहेब, Principal Sharad Chandra Pawar Mahavidyalaya, Lonan, Maharashtra, who has kindly accepted our invitation and graced this event with their presence. We are also honored to have among us our Milin sir. I extend my heartfelt welcome to all the invited speakers, delegates, faculty members, research scholars, and students who have actively participated and contributed to the success of this conference. As we gather here to bring this academic journey to close, let us take a moment to celebrate the collaborative spirit and scholarly achievements that have emerged from this event. May the deliberations held here inspire further research and action toward the conservation of our planet's precious biodiversity. Once again, a very warm welcome to Principal Dr. Chandrakant Khilari Sahib and all. Thank you for being part of this meaningful and memorable occasion. Thank you. Over to Ankita.
Thank you, sir. Now I formally invite Dr. S.B. Davange ma'am to introduce our valedictory speaker, respected Principal Dr. C.J. Khilare, sir. Over to you, ma'am.
Thank you, Ankita ma'am. It is my honor to introduce a distinguished educator and leader in the field of academia, Dr. Chandrakant Jagannath Khilari sir. He is currently working as the principal of Sharad Chandra Pawar Mahavidyalaya, Lonan, Satara. He holds a BSc, MSc, PhD and PhD Botany from Shivaji University, Kolhapur, showcasing his deep commitment the field of science. Sir, with over 36 years of teaching experience, including 25 years in postgraduate and research, Dr. Khilari sir has also dedicated 19 years to administrative leadership across various colleges, significantly contributing to the Rayat Shikshan Samstha Satara network. His expertise span mycology and plant pathology, fungal diversity, biofertilizers for agriculture, and plant conservation and management, making him a valuable asset to the academic community. Dr. Khilari sir has received numerous accolades, including the state level Ideal Principal Award in 2023 and Honorable Shankarao Kolle Ideal Life Worker Award in 2022, reflecting his outstanding contribution to the education and leadership. He has published 45 research papers, holds 5 patents, and guided 6 PhD students, further establishing his reputation as a leading figure in scientific research. Dr. Khilari sir unwavering dedicating to empowering students and enhancing the quality of education is truly inspiring. I would like to request Dr. Khilari sir
Sharad Chandra Pawar Mahavidyalaya, Lonan, Satara, to deliver the valedictory speech for today's international conference on advances in life science, biodiversity and conservation. We look forward to gain valuable insights from you, sir. Welcome, sir.
Thank you. Namaskar and very good morning. Sorry, and very good evening to all of you. Honorable Principal Dr. Sarodeswar, the faculty members of SHGM College, Kopargaon, the students, distinguished invitees, guests, and most importantly, the participants, ladies and gentlemen. I am pleased to be amongst this August gathering on the occasion of the International Conference on Advances in Life Sciences, Biodiversity and Conservation, organized by the Departments from Life Sciences and Internal Quality Assurance Cell of SHGM College, Kopargaon. I am grateful to the college authorities for inviting me for the chief guest of valedictory function of this conference. At the outset, I would like to extend my hearty congratulations and very best wishes to the delegates, the research scholars, the students who are participating in this event. Let me, at the very outset, put my record on congratulations to the entire Department of Life Sciences, including botany and zoology, and QAC, for playing an important role in organizing such event. I'm well aware that the college has been showing outstanding performance in education, research, and extension, which is testified by National Assessment and Accreditation Council Bangalore and college bag A++ grade. I compliment the institute for their contribution in making it one of the foremost institute at national level. I also compliment Principal for his dedicated efforts in bringing new dimensions to enable college at a during last 250 years. There were 3 industrial revolutions took place. The first industrial revolution was happened in 1784 when there was invention of steam engine. We started the development when we invented the steam engine. The second industrial revolution took place in 1923 when there was invention of electricity. Then our development was geared. And the third industrial revolution, it was occurred in 1969 when the computers. And as you know that there is the fourth industrial revolution. It took place during 2010 to 2014 when there was the use of artificial intelligence, blockchains, data science, Internet of Things, and cloud computing. Dear friends, India achieved remarkable growth in the food grain production from mere 51 million tons in 1950-51. It goes up to 316.4 metric ton in 2021-20. Horticulture, where we stood second in the world. The production of horticultural products was 103 metric tons in 1950-51. Goes up to 196.2 3 metric tons in 2021-20. We occupy the second position in horticulture. In milk and milk products, we occupied first position in world, and we got 209 metric tons of milk production during 2021-22. In fish production, we occupy second position in world, where we find 14.2 metric tons in fishes. It requires the skilled human resources that lead to generation of new technology, their refinement of dimensions, sound government policies, and high receptivity of farming community. I have given the information about the development in food grains. Though we have succeeded to produce a huge quantity of food grains, there are some challenges also. And these challenges included the productivity growth declines in the irrigated agriculture, which matters of serious concern. The second challenge is, or the serious concern is limited availability of water. Per capita availability of water is going to decline to almost 1/4 of availability of water in 1951. Another challenge is the development of bio-fortified varieties in sustainable elevation of malnutrition. It is a great challenge before us because day by day our population increases. Now is reach of 145 crores. The cultivated area is at the same time our cultivated field declined due to the industrialization, urbanization, as well as development needs. So, production of biofortified varieties in sustainable elevation of malnutrition, it is a big challenge before us. During 2020, the government of India has dedicated 17 varieties of 8 crops to our nation. India requests to UNO, United Nations Organization, to declare the year 2023 as an international year of millets. The researchers established some startups in millet production. There are various chances to establish startups in millets. The millet research and development in Hyderabad supported more than 100 startups in minor millets. Because millets, they are gluten-free and it is substitute to wheat also. As you know that millets are gluten-free and preferred sustainable for wheat. It contains 7 to 12% of proteins, 2 to 5% of fat, 65 to 67% of carbohydrates, and 10 to 12% of fibers. The main millets of India is jawar, bajra, and ragi. Millet helps to lower the type 2 diabetes as well as jawar contains the slowly digestible starch, fibers, vitamin E, phenolic compounds, and tannins. Why I am narrating these things before you? Because once upon a time, every common person from our country, they were using the millets as a staple food. But now, the Green Revolution and National Food Security Act 2030, the people get 5 kg wheat and 3 kg rice and rupees 2 and 3 respectively. This rice and wheat, they replaces millets from our plates. So there is almost a necessary to do something about the middle of the common peoples. It is a big challenge before the peoples from life sciences. Another thing I would like to bring to your kind notice that being a student of life sciences, it is our duty to see some things in our life sciences which will be bring some innovations for the common people. Innovation in food processing sector. Food processing industry depends on agriculture produce. Indian population is around 140. There are 33 crores of families resides in India. Or if we consider 4 members per family, the requirement of one family is rupees 3,000 as food expenses. India still spends rupees 12 lakh crores per year on food. So, size of Indian food industry of minimum scale of rupees 1,200 billions per year. Hence, to cope up with the need of food, we need to bring some innovations in food sector through the by means of some innovation methods. So innovation in food industry, technological innovation with social and cultural innovations too. Another one is smart food sector. The evolution of synthetic meat, it is also one of the alternative for the food purpose. The extraction of nutraceuticals from seafood processing waste as well as natural and potent in today's era. So these are various avenues which includes under the life sciences. We can bring some things, we can invest or to do some research work so as to we can provide the good food to people. Dear friends, the whole world is looking at it and the innovations being done by the Indians. Maharashtra has opportunities in agribusiness, food processing, and post-harvest technology, tourism, as well as renewable energy. As far as unicorns and startups in India is concerned, it has over 70 unicorns, provides unique ambience to the younger generations to thrive and succeed in life. Over the next decade, India will have the highest population of young people in the world, more than 50% below the age of 35 years. Today's technology and medicine working together. It is very important today. If you look at many of the for example, the cause-effective relationship, for example, sugar causes diabetes, smoking causes cancer, drinking spoils the liver. All this cause-effect has been found by engineers working closely with doctors. So our engineers who know medicines or medical doctors who know about engineering. So there is a very important intersection between the engineers and medicine, and that is going to be very important for quality healthcare. If we look at the Nobel laureates, 37% of Nobel laureates are physician scientists who have understood both medicine and engineering. When India was struggling for ventilators during COVID-19 period, we did come up with indigenous to solve the problem. It was required a strong interaction between engineers and medicines. It is also satisfying to note that various universities, institutions, and colleges have released several varieties and technologies for the direct benefit of farming community. They are also working in the contrast area like climate resilience agriculture, nanotechnology, biotechnology, pesticide residue testing, post-harvest technology, high-tech horticulture, etc. They have taken initiatives on innovation, entrepreneurship, startups, but the poor across the world have been repeatedly thrown into crisis management, dealing with one disaster after another. It may be COVID-19 or climate-related emergencies like drought or floods, and they are worstly affected. I would like to bring something before you about the biodiversity. The global biodiversity, I think much more discussion have been done on this aspect. It may be. Global biodiversity includes more than 99% of all species. Like current species range from 2 million to 1 trillion, but most estimated around 11 million species or fewer. With only 2.5% of world's India accounts for 7.8% of recorded species of world, including 45,000 plant species and 94 recorded animal species. There were 34 globally identified biodiversity hotspots, including Himalaya, Indo-Burma, Western Ghats, and Sri Lanka's Sundar land. It's home to 4 of the 34 global biodiversity hotspots and focuses threats like habitat destruction and over-exploitation. India has a rich flora with over 45 plant species, including 12,095 endemic species. India's fauna includes 91 species of animals, where it home to over 500 species of mammals, 200 species of birds, 30,000 species of insects, around 2,500 species of fish, and 400 species of reptiles. When we see the figures of the there is utmost necessary to conserve these entities as well as this treasure. So recent advancement in biodiversity conservation, the emphasizing the pivotal role of technology in addressing the challenges posed by climate change and the increasing threat of various species. The technological marvels such as the artificial intelligence, remote sensing, and that have revolutionized biodiversity conservation. Artificial intelligence for wildlife monitoring and species identification. The satellite imagery for forest and green. The use of artificial intelligence in wildlife monitoring and like spatial monitoring and reporting tools, as well as the protection assistance for wildlife security. We can use the technologies for monitoring the species as well as for their identification. The use of artificial intelligence in species identification by naturalists, that the citizens use artificial intelligence to help identification of plants and animals also. There are some genetic technologies like CRISPR-Cas9, DNA barcoding of live projects in DNA, which will collecting and analyzing DNA from the environment. That is from one population. To the presence of species, particularly used for the aquatic biodiversity. So the conservation tools like the biobanking, the genetic sequences is also playing a great role in the conservation of these traces. Dear friends, I would like to bring your attention towards the another thing which is too much related with the life sciences, that is the green hydrogen mission. The hydrogen mission, that is India will become a production and export of green hydrogen. Union Cabinet has sanctioned rupees 19,744 crores, that means $2.3 billion, to produce 5 million metric tons per annum with energy of 125 gigawatt. Hydrogen produced through water, it requires the electrolyzation. The water, for the electrolyzation, it requires the electricity. We got the hydrogen in various forms. There are various types of hydrogen: the green hydrogen, the gray hydrogen, the black hydrogen, and the brown hydrogen. Green hydrogen source is absolutely from water. Gray hydrogen from the combustion of diesel and petrol. The black hydrogen, obviously, it is from the coal, and the brown from the reactions of gasification. It is expected to save $12.5 billion from fuel imports, averting 15 million metric tons of annual emission of carbon dioxide. So it is a huge investment to the tune of rupees $1,000 billion. While the production of hydrogen, there are various challenges. These challenges are electrolyzers, the availability of electrolyzers, energy sources. The end use it is quite difficult, and the end dangerous resources. These are four issues which creates the challenges for the production of green hydrogen. For production of 1 kg of hydrogen, it requires 39 kilowatt of electricity. Then it requires the electricity. For the production of electricity, it requires the coal. That means we have to utilize our natural resources. So production of electrolyzers like nickel and platinum, earth metals like lanthanum and zirconium. So hydrogen is highly combustible and it is volatile. For the production of 1 kg of hydrogen, it requires 9 liters of water. This is the eco-friendly project, but then it will create some pollution resources. That means it will absolutely create a burden on the biodiversity of our country as well as the plant biodiversity. So the fraternity from the life sciences, they have to think over that. The researchers who are involved in the biodiversity, the life science researches, they can do such kind of researches which will helpful to the common persons. They are going to solve some problems of the common people. So this is the duty of each and every researcher to find out a way so that our common people will live luxuriously. Once again, I would like to thank to the principal Dr. Sarode and the organizers that they have given me an opportunity to deliver a valedictory talk. And I'm also thankful to all the participants who are joined on online mode and they have listened to me. Thank you very much.
Thank you, sir.
Thank you, sir. Now I take this opportunity to propose a heartfelt gratitude to Dr. C.J. Khillare, sir, for delivering the inspiring valedictory speech. Your words of wisdom and encouragement have added great value to this event. We are truly grateful for your presence and contribution. Now I invite interested participants to share valuable feedback on the conference. You can comment now.
Hello.
Anyone
Am I
interested
audible?
can comment now.
Am I audible?
Yes,
Hello.
yes.
Good evening, everyone. My name is Shantanu Arote. I would like to express my heartfelt appreciation for the one-day International Lean Conference on Advances in Life Sciences, Biodiversity and Conservation. Special thanks to Honorable Principal M.T. Sarode sir for warm welcoming address and Dr. B.K. Maske sir for their insightful inaugural speech. I particularly enjoyed the engaging discussion during the intellectual section. I was especially moved by the presentation of Professor Dr. S.R. Yadav sir on biodiversity conservation. His dedicated efforts in conserving numerous plant species, including various palm species at Shivaji University, is standing as an impressive and commendable achievement in the field of biodiversity conservation. Overall, the event was exceptionally well organized and intellectually stimulating for me. It was a pleasure to be a part of such a meaningful academic gathering. Looking forward to more such enriching events in the future. Thank you once again to everyone.
I request if anyone is interested to add upon the feedback. Okay, thank you. Now I would like to gently remind those participants who have not yet completed the feedback form to kindly do so. The form has been shared in the WhatsApp group for your convenience. Before moving forward, I request all the participants to please switch on your camera and mute your mic for a minute to capture screenshot. Now, I would like to request respected Dr. N.V. Malpure sir to present the conference report.
Malpure sir, 1 minute.
Thank you, madam.
Malpure sir, 1 minute.
Good afternoon, everyone.
Hello. Dr. Ravindra Chaudhary sir.
Am I audible?
Yes, sir, yes.
Good afternoon, everyone. Myself, Dr. Nilesh Malpure, convener of this international e-conference, presenting the report of today's international e-conference on advances in life sciences, biodiversity and conservation. Under the dynamic leadership of Honorable Principal Dr. Madhav Sarode Sahib, Rai Shikshan Samstha's SSGM College, Kopargaon, Department of Botany and Zoology, under the aegis of Internal Quality Assurance Cell, successfully organized this international e-conference on 8th April 2025. This conference was divided into different sessions, which includes inaugural ceremony, keynote session, 3 plenary sessions, 2 parallel sessions for research paper presentations by the participants and valedictory function. Total 58 abstracts were received for this conference and total 195 academicians and research students from more than 6 different states attended this e-conference. All the participants enjoyed the lectures, technical sessions as well as presentations. Conference started with welcome speech by Honorable Principal Dr. Madhav Sarode Sahib. He briefly introduced the chief guest and guest of honor to the audience. After welcome speech, Honorable Advocate Bhagirath Shinde Sahib Vice Chairman of Rayat Shikshan Samstha Satara, gave his best wishes for this international e-conference. The conference was inaugurated by Chief Guest, Honorable Vice Chancellor, Professor Dr. D.K. Mhaske Sir, Karmavir Bhaurao Patil University, Satara. He stimulated the audience with his very influential speech and enlightened everyone by sharing the valuable knowledge about recent trends in life sciences. Inauguration ceremony was followed by keynote address by Professor Dr. Dipti Yakanwala, Senior Professor at Department of Botany, University of Peradeniya, Sri Lanka. She talked on unraveling regional biodiversity through molecular markers, an effective strategy. She highlighted about the cross-country collaborative research work in is a need to solve many taxonomic problems. The first intellectual session was started with the talk by eminent taxonomist Dr. S.R. Yadav, INSA honorary scientist at Department of Botany, Shivaji University, Kolhapur, who talked on role of educational and research institutes in documentation, conservation, and utilization of plant diversity. Sir has talked about the important role that can be played by educational institutes in conservation of plant diversity. During second intellectual session, Dr. Shakti Kumar Singh, Scientist D, Ministry of Environment, Forest and Climate Change, New Delhi, delivered a talk on importance of biology in relation to vector-borne diseases and horticultural crop pest. The third intellectual session was conducted by Dr. Palatti Alessinu, Scientist C, Central University of Kerala. He discussed on the plant-pollinator interaction for food security, and he highlighted the importance of the uh, pollinators which are helpful for the food crops. The fourth session was of research paper presentation. This session was divided into two separate slots to give opportunity to all the researchers to present their data. Slot 1 was chaired by Honorable Dr. Milind Shinkhede Sir, Associate Professor and Head Department of Zoology, Sindhu Mahavidyalay, Nagpur. And slot 2 was chaired by Ms. Sheetal Londe, Department of Botany, SSGM College, Kopargaon. Total 38 research scholars presented their research papers. Some of the presentations were really to be mentioned, such as presentation by Ms. Monali Kadu, Ms. Aditi Saravgi, and Gitika Sukaramani. The chairperson of the session highlighted in details the importance of research in life sciences and concluded the session. Total 38 papers are received for publication, which will be published in International Journal of Biological Sciences, Biology Sciences with ISSN 2664-9934, which is published by Comprehensive Publication, New Delhi. Oral presentations were conducted in two parallel sessions. This was followed by valedictory function. Honorable Principal Dr. Madhav Sarode Sir welcomed the gathering. The chief guest for the function was Principal Dr. C.J. Khilare Sir, Sharat Chandra Pawar Mahavidyalaya, Lonan, Satara. During valedictory address, Principal Dr. Khilare Sir shared the valuable knowledge about the challenges and about the life sciences and also highlighted the importance of millets in daily diet. Some of the participants, to mention Mr. Shantanu Arote, he expressed his view on this e-conference. The participants appreciated the deliberations taken place during e-conference and also appreciated the planning, coordination and management of this e-conference through the feedback. Now we are in valedictory function, and at the end of the report, on behalf of our college and all organizing team, we thankful to the chief guest, resource persons, eminent faculties who were present online from their hectic schedule and presented their findings before us. We are thankful to our Honorable Principal Dr. Madhav Sarode Saib, Vice Principals, all head of the departments, all teaching staff, non-teaching staff, our technical team who had helped us to make this event successful. We are also thankful to all the participants from various colleges who were present virtually. Some of them presented their research work and made the event successful. We hope you might have enjoyed the invited talks and presentation as well. So once again, I thank you all for your patience and your presentations. Thank you very much. Over to you, madam.
Thank you, sir. Thank you so much.
Ankit,
As we draw close. Closer
Ankita,
to the culmination of the conference, I now have delivered the formal vote of thanks. Her words will echo our collective appreciation to all those who made this event grand success. Now I hand over the session to you, ma'am.
Hello. Ma'am, 1 minute. Dr. Ravindra Chaudhary, sir, BOS Chairman of Zoology, Savitribai Phule Pune University, Pune, and Vice Principal of Shivaji College, Jundar, is here with us in these valedictory functions. Thank you very much, Dr. Chaudhary, sir, for your presence here. Thank you. Okay, Ankita, ma'am, continue.
Yes, sir.
Continue.
Now
Proceed
I
for the vote of
request.
thanks. Proceed for vote of thanks.
Now I request SS Gosavi ma'am to deliver the formal vote of thanks.
Thank you, madam. Good evening to everyone. It is a great honor and privilege for me to propose vote of thanks on behalf of the organizing committee of today's one-day international e-conference on advances in life science, biodiversity, and conservation. First and foremost, I express my heartfelt gratitude to the chief guest of today's inaugural session, Honorable Dr. D.K. Miske Sahib, Honorable Vice Chancellor of Karmar Bhaurao Patil University, Satara, for his gracious present and inspiring words. I extend my sincere thanks to our keynote speaker, Dr. Dipti Yakandwala Madam, for her enlightening and thought-provoking address that set the perfect tone for the day. I am especially grateful to my idol and our eminent speaker for intellectual session first, Dr. Yasar Yadav sir, for his beautiful presentation and continued inspiration to generations of taxonomists and botanists. My sincere thanks also goes to Dr. Shakti Kumar Singh sir, resource person for intellectual session second, for his sharing deep insights and expertise with all of us. We are equally honored to have Dr. Paleti Alice Sinu sir as the eminent speaker for intellectual session 3. Thank you for your valuable contribution and time. I extend a warm note of thanks to the chief guest of today's valedictory function, Principal Dr. C.J. Khilari Sahib, Principal, and for his inspiring words during the closing ceremony. I have heartfelt thank you to Honorable Advocate Bhugirat Kaka Shinde Sahib, Honorable Vice Chairman of Rai Shisha Sanstha Satara and Chairman of College Development Committee, for his constant support and encouragement throughout the planning and execution of this event. I can also take opportunity to thank all the members of College Development Committee for their support. I am very thankful to our dynamic principal, Dr. M.T. Sarode Sahib, chairman of the organizing committee, whose visionary leadership and unwavering commitment have been instrumental in the steering of SHGM College towards consistent academic excellence, and to all our vice principals for various faculties for their guidance. A big thank you to all the journalists who covered this conference. Thank you, our teaching and non-teaching staff of SHM College for their contribution and cooperation in making this event successful. Special thanks to our conference coordinator, Professor H.T. Mate sir, Head Department of Botany, and our organizing secretary, Dr. V.R. Jutode sir, Head Department of Zoology, for their tireless effort and excellent planning. I also extend my gratitude to our convener, Dr. N.V. Malpuri sir, IQSC coordinator, for his support and constant motivation throughout the conference. Thank you to all paper presentators and participants of the conference for sharing your research and being an active part of this academic platform. I deeply appreciate the efforts of all organizers and technical team whose behind the scene dedication ensured the seamless execution of this international e-conference. And of course, our helpful thanks to our wonderful anchors, Dr. Davange Madam and Ms. Prasad Madam, for elegantly conducting the today's proceeding with grace and clarity. As we conclude to today's academic celebration, let us take forward the knowledge and inspiration gained here to further our efforts in research,
conservation, and collaboration. Once again, thank you for all being part of this enriching experience. Thank you very much. Best wishes to everyone. And with permission of our chairperson, Principal Dr. C.J. Khilari sir, I now declare this meeting is completed.
Okay, Khilari sir.
Hello. Thank you, madam.
Okay, mute it. Now close this.
Thank you. Thank you, everyone.
Thanks all.